Pablo again for his contribution. I'm sure we can catch up during lunchtime. So the next speaker is going to be Professor Marco Garcia-Fuentes from the Universidad de Santiago de Compostela, and he's going to present about bone morphogenetic protein 7 loading microsphere can suppress glioblastoma initiating cell in human primary tumor model.
Yeah. So thank you very much. I would like in this presentation to show you a little bit of our latest data regarding the management of glioblastoma. So glioblastoma is the most common type of brain cancer, and contrary to many other tumors in which care has advanced, treatment of glioblastoma is still done by resection and treatment with temozolomide and radiotherapy. And although this protocol has improved probably 3 or 4 months the average life expectancy, the survival rate at 5 years of glioblastoma is still around 5%. And right now we have a conceptual framework why we think this might be occurring. And conventional, so it's known now that many solid tumors might have a subpopulation of cells that they are sometimes called cancer stem cells, sometimes they are called tumor-initiating cells. These cells tend to be hyperresistant to chemotherapy and hyperresistant to radiotherapy. So when you are managing a tumor like glioblastoma through conventional therapies, you usually just remove the non-cancer stem cell types, and you leave behind the cancer stem cell cells that will be able to rebuild in a few months the tumor de novo. So actually, the kind of therapy that we would like to have is exactly the opposite. So some kind of material or some kind of drug that will be able to deplete the cancer stem cells, and because then the non-cancer stem cells will be able to resolve probably just by itself or with conventional therapy. So we are interested in materials that are capable of modulating stem cells. So when we saw this perspective, we thought we could do something about it, and we were very interested in the work of Giloblastoma, that he was really analyzing the pathway of bone morphogenetic protein. This is a protein that is mostly used for tissue engineering of bone, but it actually happens to have a lot of activity also in the brain. And in the brain, it is known that it might be able to transform cancer stem cells into non-cancer stem cells. That would be a differentiation therapy. However, we understood that if we wanted to use that bone morphogenetic protein in the brain implanted after resection of a tumor, we'll need to have an appropriate delivery carrier, because the half-life of this protein in the brain will be just probably 5 minutes. So we wanted to have something that will be able to provide regional delivery to the tumor and control release. So have this protein for enough time to be able to transform or reprogram all the cells that are cancer stem cells into a more benign phenotype. And so for that, we decided to integrate this into a biodegradable matrix that we were intending to implant once we remove the tumor from the brain. But of course, proteins, they are difficult to integrate in biodegradable matrices because they don't have much affinity for many of the hydrophobic polymers that they use. So there are many ways to resolve these issues. In our case, we patented a technology. This technology is based on the affinity that these kind of molecules have for heparin. And you will form a complex, and then we form over this complex, we coat it with another compatibilizing polymer. In this case, it's a tetronic. And we create basically a nanostructure that we can then very easily integrate in a biodegradable matrix. We thought that for our intended application, which would be just implantation after the resection of the tumor, it would be most interesting to make it in the form of a microsphere. So these are the microspheres, and you can see that we can make them between 20, 40 micrometers in size. And due to the procedure that we are using, we are able to generate more than 90% encapsulation of the BMP. And actually, this is a value that is higher than the encapsulation obtained by double emulsion, which is the standard method for encapsulating protein in this kind of materials. So we're able to see that we had over 2 months of sustained release of the protein in vitro, and we were able to correlate the sustained release with the biodegradation of the matrix. So we wanted then to know whether this BMP that is released after 1 month or 2 months or 3 months is still bioactive. So what we did was to generate a special kind of culture when we generate or we use a cancer stem cell isolated and we grow it into the form of a tumor. And we saw that when we grow these tumors in a plate, under controlled conditions, we will form a number of tumors. But if we put some of the released BMP-7 in the plate, we will be able to suppress the formation of many of these tumors. So we knew with that we had an active drug, and we decided to move ahead, and we got 8 primary cell lines from human glioblastoma, and we decided to test these kind of therapies in vivo in mice. Basically, the experiment would be that we seeded some primary glioblastoma cell lines together with either blank microparticles or BMP-loaded microspheres. And we implanted this into the back of mice, and we wanted to see what was the activity, right? So basically, we already observed that tumor formation was, tumors were formed in the control group, so the empty microspheres. After 2 months, the primary glioblastoma cell lines were forming the tumors. And we tested 2 doses for the BMP-7 loaded microspheres. We saw that for the lower dose, there was not a very clear effect, but when we used the higher dose, it was very clear that the mice were generating smaller tumors. And that was also confirmed when we resected the tumor from the back of the mice. And this is actually the quantitative data, so we could see that very similar tumor volume for the blank and the lower dose microspheres, but smaller tumor size for the higher dose, and this is the same but final tumor volume. Well, interestingly, of course, this is a non-cytotoxic kind of therapy, so we wanted to check in the histology whether we will see any kind of cytotoxic morphology in the cells, and we could see or confirm that there was no toxicity in the cell. So, of course, one of the points that we are defending with this way to address cancer stem cells is that sometimes cytoreduction or reduction of tumor volume will not translate well to survival values. We think that there need to be sometimes other kind of endpoints that we need to analyze. And we were even more happy that with these results, with these other results, we show many biochemical markers of the tumors and how it changed when we use the blank microspheres or the BMP-loaded microspheres. And we saw, for instance, that many traits that are associated to cancer stem cells, like, for instance, CD133 and OLIC2, which are typical of cancer stem cells, they were reduced. Pro-proliferative markers like GFAP, delta, and PCNA were reduced also. These are very typical for malignant tumors. And cell block proteins like these 2, which are cell cycle inhibitors, they were very, very clearly upregulated. So in general, the tumors resulting from this therapy have a more benign phenotype than the original tumors. So basically, we were able to generate or confirm that we have a technology with clear preclinical efficacy. We think that this therapy might be used mostly after tumor genomic classification, and probably together with adjuvant therapy, probably for further cytoreduction or with immunotherapy kind of drugs. So just acknowledgements, this is the funding bodies, and the biomaterials and the strategic part was performed at the University of Santiago de Compostela for Research Center by these people. And most of the in vivo work was done in the Instituto Salud Carlos III from this team of researchers. Thank you very much for your attention.
Have we any question, please?
Thank you for a very nice talk. Actually, it's a very interesting concept. I have one question on your in vivo model. Did you look for parasite coverage? Because BMP-7 is inhibitor for parasites. Parasites are like mesenchymal cells, and BMP-7 is basically anti-fibrotic growth factor. And it can inhibit the fibroblast activity, but also in these models, they're hardly fibroblast, they are more parasites. And how can you say whether you inhibited cancer stem cells?
Yeah, okay. Regarding the first question, we have not tested all the kind of phenotypes. From the histology, we have looked to the vasculature a little bit, and we think it's more or less normal. We cannot be sure 100% whether all parasites are at the same level, but more or less normal. Regarding cancer stem cells, well, what we know is that several cancer stem cell markers in vivo, they are downregulated. So we think we are suppressing these traits from the tumor.
Thank you very much.
Okay, so if you agree with me, we have one last speaker that divides us.